Hello everyone and welcome to today's video. In today's quick video, we're going to be talking about chromatin, its types and functions. So first of all, before we're getting and diving deep into it, we have to understand what chromatin is. So essentially, chromatin is a complex of DNA and proteins. And these proteins could be histones or non-histones. Regarding its functions and why is it important, first of all, to package the DNA in a more compact shape. And also this is extremely important because I guess in future videos, we're going to dive into more the different levels of a compaction that can be found inside the cell, or inside the nucleus. Second, to reinforce the DNA macromolecule to allow mitosis. Third, to prevent the DNA damage. Here, I'd like to give an example where the proteins and the histones here and the non-histones essentially function as cushions for the DNA to prevent its damage. And fourth and finally, to control the gene expression and DNA replication. I really want to stress on the importance of this point. And here we can say that, as we're going to be seeing in the next slide, that there are certain regions where the chromatin structure is relatively loose, allowing the gene expression to be taking place. And here we're going to be talking about the first type of division, and this is euchromatin. So essentially, in these regions, there is going to be compaction. There is going to be this package between DNA and proteins, histones and non-histones. However, it is going to be relatively loose, allowing the genes that are present within these regions to get expressed. Whereas when we look at the other type, which is the heterochromatin, essentially these are regions where the packaging is more compact. And this is going to be preventing the expression of genes that are present within these regions. Now, it is worth noting that within heterochromatin, there are two major types: the constitutive heterochromatin, and here we're going to be talking about areas where this dense structure, this compact structure of a packaging between DNA and proteins is going to be like this the whole time. And here we're going to be referring to areas such as the centromere. And the second type, which is the facultative, these are heterochromatin regions that are indeed dense, that are indeed that are packaged compactly. However, they can also transform into euchromatin if the right signal is given by the cell. So here, essentially, these are regions where there's compaction and there's no expression of the genes that are present within these regions. This is oversimplifying this. However, there's no expression, and when the right signal is given, this is going to be turning into euchromatin, facilitating and allowing the expression of genes that are present within these regions. This concludes this video. Thank you so much for watching. Don't forget to subscribe, like, and comment. This has been Ali from B.Biology.